All right, guys, let's talk a little bit more about human medical genetics, and we'll begin with an example, a bit of a case study. So I'm going to talk about type 2 diabetes now. Type 2 diabetes is pretty common. About 1 in every 10 people in America have this disease. About 25% of those people don't know they have it, because in the early stages, you're basically symptom-free. Almost triple that number, about 1 in every 3 Americans is pre-diabetic, which is elevated levels of blood glucose, but not elevated to the point at which you would be considered a diabetic. And this disease is incredibly expensive for us as a society. About 25 billion a year, I believe, is what it costs. So why might this be the case? Well, and why might those numbers be so high? Well, this is just interesting to look at. So let's look at this. Here are 2 maps of the United States. On the left, levels of obesity in the United States, and on the right are levels of diabetes. And essentially, the darker the color, the higher the number. So the darker the color on the left, more obese people live in that state. The darker color on the right, the people in that state have a higher incidence of diabetes. And what you see, looking at these 2 maps, is that there's some kind of correlation, right, between obesity and diabetes, generally speaking. Now, this map is from 1994. And what I'm going to do now is let the maps tick over year by year over the upcoming years. And we're going to watch how they change. So 96, 97, 98. And what you can see over time is that both maps are becoming darker and darker. And you can see again that there is a correlation between the darker areas in both maps, suggesting that obesity and diabetes are related. And that is indeed true, because obesity is the environmental risk factor for this disease, right? And you can see over the last 20 years or so, as a country, we've got a lot heavier and we have a lot more diabetes. The reasons behind that are myriad and have been speculated about endlessly, but I'm not going to talk about those. I'm just going to talk about the disease itself. So 2015, the last year that this kind of data is available, you see that a lot of very overweight people and a lot of people with diabetes, and they correlate, generally speaking. About 1 in 4 of us will get type 2 diabetes in our lifetimes. I am one of those people. I'm a type 2 diabetic. This disease is very complex. It's one of those complex polygenic diseases. And there's also a big interplay between environmental and genetic factors. And so when you're thinking about genetics and its ability to predict whether or not you will get a disease, you have to think about whether or not you would want to know what your risk is going to be. And when we talk about any genetic disease, then this little equation gives you a framework with which to think about whether you want to know the answer to the question of your personal risk. And the desire to know is risk times by burden times by intervention. Risk in this case is how likely am I to get the disease? Am I 90% likely or 9% likely to get the disease? Burden is how bad is the disease? Is it something like Alzheimer's disease, which has a very high burden for both you and your family? Or is the disease more like, you know, tennis elbow or something? Not a very high burden for you and your family. Last but not least, intervention. Is there anything I can do to help myself either not get the disease or intervene if I do get the disease? So again, Alzheimer's right now at least has 0 intervention, basically. So what we have to consider when we're thinking about how to think about human genetics is 2 types of risk. absolute risk, which is the overall chance in the population of getting a disease. That's that's sometimes called the baseline risk. And then there's your personal risk relative to that average person. And this is how you're given odds of getting a human. This is how you're given the odds of getting a disease based on your genetics. So if your relative risk is 1, you have a risk that is the same compared with the average person. If the number is above 1, so 1.2, for example, you have a 20% higher risk than the average person. And you can also have protective alleles that lower your risk for a given disease. As we're using type 2 diabetes as our kind of case study here, the absolute risk, so the baseline risk in the US at the moment of getting this disease is 23% by the age 60. That's just the numbers in the population. The strongest genetic risk allele for type 2 diabetes is a gene called TCF7L2. It's actually a transcription factor. Make of that what you will. And it has a relative risk factor of 1.4. So if you carry a certain allele of this gene, you have a 40% higher chance than the rest of the population of getting this disease. So you can work out your relative risk by taking the absolute risk, 23%, multiply it by your relative risk if you have the allele, which gives you a 32% chance of getting this disease. So that's how you basically factor in genetic risk to your probability of getting a disease. Now, genome-wide association studies have actually identified many risk alleles for type 2 diabetes, more than 100. But how do they interact? It appears as though, although I think that it is fair to say that we don't, we need more data to say this with a fair degree of certainty, that different alleles act independently. So if you carried more than one allele to getting type 2 diabetes, they don't act in a sort of in a compound manner. So that's how you calculate the risk of getting a disease. Burden, if you're talking about type 2 diabetes, as our case study, it's the 7th leading cause of death in the US, has all sorts of phenotypes that are not fun. So the burden is pretty high. What about the intervention for type 2 diabetes? Well, it's highly preventable based on your interaction with the world, because the environment has a big impact on whether or not you get this disease. Obesity, as we saw earlier, but also diet and exercise play a big role in whether or not you get this disease. So the question is if obesity is a contributing factor, how much weight do I have to lose? This is what you get, by the way, if you Google topless man and do an image search. You get this very in-shape dude delivering bacon out of a hotel room. It's very confusing to me. But do I have to look like this guy? Because I do not look like this guy at all. So actually, no, the studies that have been done into type 2 diabetes, the initial study suggested that losing 7% of your weight loss had a huge impact on whether or not pre-diabetics were going to develop fully fledged type 2 diabetes. Further studies actually suggested that you can reduce your risk of getting type 2 diabetes by losing as little as 5 to 10 pounds, no matter your starting body weight. So in this case, I think knowing whether or not you are predisposed to type 2 diabetes is probably a good thing, because it has a high burden, but it also has a very strong intervention. However, what that really means, right, is that if the intervention for type 2 diabetes is to basically eat better and exercise, don't you know that that's good for you already, right? So shouldn't we all be doing this anyway? And of course, the answer to that is yes. By the way, pretty much the only thing that's shown to have a protective effect against Alzheimer's disease is staying in shape as well. That's true for a bunch of cognitive diseases. But one way genetic testing can sort of help inform the way that you live your life is whether or not you would be more or less likely to live more healthily if you knew you had a higher genetic risk for a disease. Are you of the opinion that, hey, I'm more predisposed to this disease, therefore I'll work out and do my cardio? Or are you more of the mind that, hey, I'm going to get this disease anyway, so, screw it. So this idea of having genetics and having it play a role in healthcare led to President Obama starting the Precision Medicine Initiative a few years ago. What does this mean? Precision medicine is sometimes called personal medicine or personalized medicine. And basically, it's the idea that you can sequence someone's DNA and tailor their lifestyle and or treatment according to their genetics. So there's a couple of goals of precision medicine or personalized medicine. Learn your risk of diseases and manage your lifestyle accordingly, like in the type 2 diabetes example. And the second sort of broad idea is that it'll allow us to develop personalized treatments for patients. We already talked about risk of disease and managing your lifestyle. So let's talk about how in the future, and this is already actually starting to happen, you can maybe have a personalized treatment based on your genetics. So that brings me to a discussion of a newly emerging field called pharmacogenetics or pharmacogenomics sometimes. And this is that you guys all know that drugs have different effects on different people, right? Some people require more drugs to have an effect than other people do. And that's because of genetics, essentially, because drugs can have very different effects on different people depending on how quickly they're transported into your body. That's all regulated by proteins. How quickly they get to target cells, again, it's all regulated by proteins. The ability of the drug to target whatever protein it's designed to target, of course, that depends on that protein. How quickly you metabolize a certain drug in your liver. Again, all of these things are linked to genetics because they're all due to protein activity. So in the future, it is probably true that your genome may be sequenced to improve your diagnosis and care. That might be particularly true in terms of what are sometimes called off-target effects or side effects of drugs. Some people suffer from horrible side effects, some people don't. You might be able to predict that by sequencing someone's DNA. So let's watch this little video of Max and Hannah just to flesh out that idea a little bit.
This is Max.
And this is Hannah. They both suffer from an autoimmune disorder.
Max is a patient today, and his story is based on current medical practice.
Hannah's story is our prediction of what will happen in the future when medicine has become personalized for this condition.
Max's doctor correctly identifies his condition and prescribes the standard first-line therapy, corticosteroids. Max tolerates them poorly and experiences nausea and vomiting.
Max goes back to the doctor, who decides to revise his dose. However, Max still suffers adverse events.
Max goes back to his doctor, who switches him to non-steroidal drugs. The nausea and vomiting subside, but the drugs have limited efficacy. He remains prone to hyperimmune reactions.
Hannah visits her doctor and is also diagnosed correctly. She undergoes genetic and serum protein tests. These establish the genetic cause of her problem and help identify effective and well-tolerated treatments.
Diseases with the same symptoms often have different genetic causes. Genetic tests allow doctors to identify the roots of the problem and tailor treatments to each patient. Further analysis of genetic markers can aid selection of a treatment with minimal side effects.
Hannah's doctor prescribes a genetically tailored drug for her condition. The new medical technology allows the doctor to treat the patient, not just the disease.
Max suffers a hypersensitivity reaction and is hospitalized. His doctor prescribes Imuran after struggling to find a milder therapy which Max can tolerate. This leaves Max prone to cancers.
Hannah's treatment is successful and well tolerated. Her condition is managed effectively.
So as you can see, I'd rather be Hannah than Max, right? And this is definitely already happening in the clinic. The idea that people have their DNA sequenced to perhaps prescribe a better drug for them. Pharmacogenomics and these different sort of genome-wide approaches are great, but historically speaking, the kind of DNA that has been sequenced tends to be from white European people in the United States. And so the National Institutes of Health, a couple of years ago, or a year ago or so, instituted a new program called the All of Us Research Program. And what they're trying to do is sequence a whole bunch more DNA from a bunch of different people of different backgrounds to try and diversify more the genetic information that's out there that we use to come up with certain tests. So hopefully, you know, with precision medicine, we can take into account your environment, your lifestyle, and your biology in terms of your genetics to treat people better. The last thing I'm going to talk about I think the last thing I'm going to talk about in terms of this is better cancer diagnostics and treatment. And there's a couple of examples that I'm going to show you of this, the way genetics can help us out in this field. And basically what you're looking at here is an individual that has some kind of cancer. And what people are doing now is taking DNA from those cancerous cells and sequencing the genome. And then they'll take some DNA from healthy cells from the same person and sequence the genome of those cells. And then using bioinformatics, right, using computers, we can compare those two sequences and see which bases have mutated in the cancerous cells versus the healthy cells. And this is useful for a couple different reasons. Firstly, it allows us to discover different, basically diagnose different cancers very much more specifically. So what is clear is that you don't just get lung cancer. That's not, the term lung cancer describes lots of different diseases that have the same appearance, tumors in the lung, but they can be due to different molecular problems in the DNA. So it allows us to accurately diagnose your cancer. Which of the 200 types of lung cancer do you have, for example? And then it allows us to target therapies to people that have that specific type of cancer. So better cancer diagnostics and treatment, that's kind of a twofer using this approach, because you might be able to figure out what type of cancer you have and what drug works best on it. The other way that personalized medicine can help you is that you can take, for example, some cells from your personal tumor, grow them up in a lab, and then hit those cells with 1,000 or 10,000 different drugs and see if any of those drugs work particularly well against your type of cancer. And this allows you to obviously try way more drugs on a cancer type than you ever could on just a patient themselves. Of course, cells grow differently in a dish than they do in the body, and that's led to cancer diagnostics diagnostics and treatment using what is called a mouse co-patient. And this is a very similar idea. This time we're taking tumor cells from your personal tumor, but we're not growing them in a dish, we're growing them in a mouse. So this mouse has a lump on its back, as you can see, that is caused by some human tumor cells growing in there. And then you can test drug effectiveness on this mouse instead of a person, and hope that if there's any nasty side effects, you find out on the mouse before you find out on the person. So that's the last thing I want to talk about as sort of personalized medicine goes in terms of actual diagnostics. But I would be remiss if I did not end this little lecture with genome editing and talking about CRISPR a little bit. So genome editing, editing the human genome. Currently, it's available. We can do it. CRISPR is this recently discovered technique that we can use to accurately edit DNA in your genome. Proof of principle is already here. So in human embryos, we've managed to correct a Mendelian genetic disease called beta thalassemia. It's a blood disease. And we even managed to eliminate a targeted chromosome using CRISPR as well. So for example, if you had if you had Down syndrome, you could potentially treat it using something like CRISPR, because you could destroy that extra chromosome. So what is CRISPR? CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a bit of a mouthful. But all you really need to know about it is that it's a genome editing technique that targets a specific piece of DNA in your genome and allows us to cut the DNA at that place. We can make a gene non-functional. We can knock a gene out using CRISPR. We can change DNA sequence using CRISPR to replace one allele of a gene with another allele of that gene. It's a very, very powerful technique. How does it work? This cartoon shows you very simply how it works. CRISPR-Cas9 is a bunch of proteins, basically, and it uses a guide RNA, as shown in this diagram in step 1. That guide RNA is basically like the targeting part of a targeted missile. By virtue of its sequence, it will bind to its complementary sequence somewhere in the human genome. That's how CRISPR is so specific. The guide RNA kind of finds where you want the Cas9 complex to go. When it gets there, it will edit the DNA as you would like it to. In this example, in step number 4, you can show that it's cutting out some DNA and it's replacing it with new DNA. So that's kind of how it works. Relatively simple, really, but very powerful. How do you deliver this into cells? This is a little movie of a microinjection technique where we're taking a needle, we're poking it into a human egg right after it's been fertilized, and then we are pumping some liquid into that egg that contains the CRISPR-Cas9 protein and the RNA and all that targeting stuff that we looked at earlier. And so this is how you can deliver CRISPR to a single cell in order to edit the DNA in that cell. Fantastic. But that doesn't help you if you're an adult. You have trillions of cells. So microinjection is considered to be the gold standard for introducing CRISPR into cells because, you're just injecting it into a cell. It's as simple as it gets. But scientists are now developing what are called different vehicles, which are just different methodologies to deliver this gene editing system to different cells. You can do it by physical delivery, such as a microinjection. You can use what are called viral vectors. So basically a viral vector is an engineered virus. You guys all know, by what's going on now, right? That viruses can introduce genetic material into cells. So we can engineer viruses to do that. And then there are other ways that are called non-viral vectors, which is a catch-all term. for different things like lipid nanoparticles that can surround the CRISPR machinery and get it into cells. And these kind of things, viral vectors and non-viral vectors, you could take as a pill or an injection, and then you could target more cells in the human body. But is this ethical? Is it something that we actually want to do? We know that we can edit the human genome because it's been done multiple times. And at the moment, they are allowing it in limited form for treatment of human diseases in the US. Watch this short video and it will explain what I mean by limited. So essentially what the NIH has done is created this thing that's called the Somatic Cell Genome Editing program. Note the word somatic. In other words, we're not going to edit any germ cells so that any CRISPR-based changes we make in DNA will be limited to the somatic cells and not the germ cells. So it can't be passed on to new generations.
Thousands of human diseases are caused by changes or mutations to the body's DNA. What if we could treat all these diseases by diving into our living cells to correct the mistakes? The Somatic Cell Genome Editing program aims to make that happen. Recently, researchers have made great progress in correcting DNA mutations using a technique called genome editing, and the first tests of genome editing for human diseases are starting. However, there are still some challenges to achieve safe and effective genome editing in patient cells. For example, the DNA in millions of cells needs to be corrected to treat many diseases, and getting the editing tool into different types of cells, such as those of different organs, will require tailored delivery techniques and packages. The delivery techniques must be specific to enter the right cells and avoid the wrong ones, such as the cells of other organs or reproductive cells like eggs and sperm. Additionally, different types of editing tools may be needed to correct different types of mutations. The Somatic Cell Genome Editing Program, or SCGE, was launched by the NIH Common Fund to develop quality tools to perform safe and effective genome editing in human patients. The SCGE program will make more genome editing tools available to researchers, develop better packages to deliver the tools to the right cells, design new tests for the safety and efficacy of genome editing, and make all of the information available to the scientific community to drive future discoveries and cures for patients.
So you can see what they're trying to do with this program. They're trying to make CRISPR applicable to treating humans in a safe and ethically, reasonable way. So the last thing I'll say in terms of ethics, just to finish on this note, is this fella. This fella is the Chinese scientist that you may have heard genetically engineered human cells and then got those eggs implanted into a real person. And that woman gave birth to these gene-edited babies. So Francis Collins, who is he's the director of the NIH and is a prominent American scientist, basically said this about the work of this Jiang Kui, Dr. that guy. He said it was profoundly disturbing and tramples on ethical norms. We need to develop a binding international consensus on limits for this research. So what did he do and why was it so bad? He took some human cells from like an IVF experiment. So take eggs from the woman, sperm from the man, mix them together in a dish. The egg gets fertilized. He took those eggs and he did CRISPR on them. And then took those eggs and put them into a woman. He then presented that data at a conference. It's never been published in a paper, and everybody freaked out. And then when we found out more and more about what he actually did, we found out it just got worse by the day. So I don't want to go into details, but he did not address an unmet medical need. There was no point in doing the CRISPR experiment that he did on these babies. He basically tried to introduce into them a mutation that would make them immune to infection by HIV. And he said that that was because their dad had HIV. And this is not a dire medical need that needs to be met. It's really hard to get HIV from your dad. So that was a bit weird. There is a known naturally occurring mutation that makes people immune to HIV, but he didn't actually execute that particular change into these girls. It's not, he basically introduced changes that we don't know what they're going to do to these girls as they grow up. There was also all these problems with informed consent. He basically did this without telling the people what he was going to do, so he operated under this cloak of secrecy, but he had this weirdly slick PR campaign. It was super weird. Everybody around the world, every scientist think it's a terrible thing that he did. And just as the capper, he actually acted in contravention of his own stated ethical views. So if you look a little bit right before these babies were born, he was a co-author he's the primary author actually on this paper, saying that genome editing was a bad idea, ethically speaking, in human beings. So it's completely bonkers. What happened to him? Well, very end of last year, he got put in jail for 3 years, essentially, because of all the ethical lapses that he performed whilst doing this experiment. So, all right, last slide of the semester. Basically, hopefully this last couple of short lectures have given you some idea of where we might be going with human genetics and treating human genetic diseases. There's a huge potential upside to pharmacogenomics and cancer treatment and those kinds of things. But we're really going to find out over the next decade or so whether or not these are going to revolutionize the world of medicine or not. So thanks for listening, and you've made it to the end. Well done. All right, guys, I hope you're doing well, and yeah, catch you later.